Okay, picking up with this final part here of section 5.4, looking at proteins. We talked about the idea of having normal proteins, that they can become denatured, and when you get denatured proteins, you get a loss of function. And in addition to getting a loss of function, you can have a lot of problems come into play. So it's really hard to predict a protein structure from its primary structure, but yet that is what determines its overall structure. Right now, we have very few ways of figuring that out well. Now, proteins do go through several stages on their way to forming this stable structure, right? So keep in mind that proteins are produced in a linear fashion, starting at the N-terminus, adding on amino acid by amino acid. So a lot of times it is folding progressively as the protein is made itself. Since we know protein folding can lead to loss of function, and we've also seen that protein misfolding can lead to larger structures of misfolded proteins, which can lead to problems. Okay, so sickle cell anemia isn't unique. Alzheimer's, Parkinson's, mad cows are all associated with misfolded proteins. So how does the cell combat this problem? The fact that you can unfold proteins and the fact that it can lead to such severe consequences. One thing that helps with that is a group of protein called chaperonins. These are essentially other proteins that can assist a protein in folding. And I will show you how one of these works, and we can talk about how some of the others work as well. Maybe we will do that in class. So here we have a chaperonin. This is one specific type, and you can see that it is somewhat barrel-shaped. Okay, you actually have 4 ring-like structures made up of smaller independent subunits. And over here we have a cap. So this actually constitutes 2 parts. This is one chamber, and this is another chamber. When you get an unfolded polypeptide, it has a lot of hydrophobic exposed regions. Just so happens that this side of this barrel-shaped structure, on the inside, you have a hydrophobic surface. So this is naturally going to attract this unfolded polypeptide. It'll want to bind to the inside of this. What then happens is you put a cap on, and there's actually a catalytic reaction that occurs, and it results in a shape change of this internal chamber. And this actually results in a shift where before the interior lining of this barrel was hydrophobic, it almost instantly switches to a hydrophilic environment. Okay? This is going to cause the protein to debind the walls, and then it's going to have to come up with its own strategy for hiding the hydrophobic regions. In this scenario, you have a single protein trapped inside of this chamber. So you don't have the scrambled egg problem of multiple proteins trying to fold on top of each other. Here, we just have a single protein separated from everything else trying to refold. That is much more achievable, okay? And we can actually result in a properly refolded protein, which can then be released into the environment. The reason why this has 2 chambers is you can actually take turns. You put a protein in this side, you put the cap on, change the shape. Over here, we can put in misfolded protein in the other side, put a cap on that side, change the shape, and just essentially have a revolving door of protein folding space. Now, how do we determine protein structure? Well, we have several techniques. One is called X-ray crystallography. This requires that we can take a protein and form a crystal out of it. If we can form that crystal, then we can shoot X-rays through it, the X-rays diffract, and we can reconstruct what that crystal must look like. There's another technique called nuclear magnetic resonance, or NMR. This is a much more complicated idea to discuss, but you can essentially find amino acids that are close to each other. And if you can find enough things that are close to each other, and I mean intimately close to each other, you can start restricting the shape and then reconstruct the overall shape. This does not require the protein to be crystallized, so you get a more natural version, but it is restricted in how big of a protein you can work with. Over here, you have to be able to crystallize the protein. On occasion, that crystallization process can actually deform the protein, and it's hard to tell when that happens. As we get more and more solved structures, we can actually start to use bioinformatics in order to predict what the structure might look like. So the more and more proteins we get, we can start thinking about, well, what is that protein related to? Do we have a structure for that protein? Can we then overlay the structures and see if it fits? So the more structures we get, the more powerful bioinformatics becomes. So that is something that is growing every day. Ultimately, our hope is to simply be able to look at the amino acid sequence and, like nature does in folding the protein, be able to model how that protein would behave and construct a model based upon that. So, concept 5.4, proteins include a diversity of structures, resulting in a wide range of functions. Proteins are made of amino acids. Those amino acids have an amino group, a carboxyl group, and a unique side chain. Those unique side chains can come in many, many different flavors. Those flavors of amino acid include nonpolar, so hydrophobic, hydrophilic. Within those hydrophilic, we could just be polar or actually be charged, either acidic or basic in nature. That overall primary sequence of a protein will lead to the formation of secondary structures. Those secondary structures can then be folded on top of themselves to provide this tertiary structure. The overall tertiary structure is mostly dependent on what those side chains are. The secondary is largely dependent on the actual backbone itself. Quaternary structure is when we bring multiple polypeptide chains together. Proteins work when they are folded properly. If you unfold it, that's called denaturization, and that leads to a non-functional protein. Sometimes it is possible to renature that protein, but oftentimes it is not possible. One thing that can help with that is chaperonates. But also remember that we start off this section looking at all the different things that proteins can do. So we talked about how the fact that it can be an enzyme, it can be a defensive protein, it can be a transport protein, it can be a hormone, it can be a structural protein, and the list goes on and on and on. So understanding how proteins work and how they fit together is a large portion of how we can understand a cell at a molecular level.